All right, Dirk, are you ready?
Ja, opname loopt al.
Okay. So what I'm gonna show you now is cross-sectional image taken in a human lung, and just to help you out with what we're looking at, I'm showing here three different images that show you the standard structural OCT information. That's this one, the birefringence, and the optic axis uniformity. But let's first look at the first image here, which I call attenuation coefficient. So there is a method to calculate how the beam is attenuated in the tissue, uh, um, like Dirk has explained when you determine the attenuation coefficient. So you can calculate an attenuation coefficient per pixel in your image. And that creates these kinds of images. So the the center here, this is in red. I delineate the plastic tube that's around the catheter, and inside the catheter is a motor that I will show you later that projects the light out like a lighthouse. But you make a circular scan, and this is the image. That's the information that you get. Um, I'm going to show you examples later as to why attenuation coefficient we think is more helpful than standard OCT images, although, that's up for debate. The second image here is the birefringence. So what we calculate is the local amount of birefringence. The birefringence is in a very thin layer. And if you look in this image that I'm showing you, there is a very thin birefringent layer right here. I'm going to circle it with my red pen. You see that faint layer of white in this image? Yeah, that's the birefringence. The third image is that optic axis orientation, where white means that in that region you have a very nicely uniform orientation of the fibers. And when it's black, it's randomly oriented in any direction. So it's the same image, and now here you can see that orientation of the uniformity of the optic axis orientation. And you can see how much better the contrast is for that layer compared to the birefringence. It's visible in the birefringence, but the optic axis really helps you to bring it out. Okay, so these are the kind of images that, oops, what happened? Jeez. Oh, it's in edit mode, okay. Okay, so we're gonna show images of the human lung. So first, what are we looking at? So we have patients that have asthma, and the difference between a normal lung lumen and that of an asthma patient is that the wall is inflamed and thickened, and you have these muscles here around the airway, and especially these muscles. Wait, there's something wrong. Yeah, especially in an asthma patient, you can have this constriction, and this gives you that shortness of breath. And it's caused by these smooth muscles around the lumen that contract. And you can imagine that if you have for an extended period these asthma attacks and these muscles contract, it's like them going to the gym. They get stronger, which makes it worse. And so the one of the treatments that they have come up with is called bronchial thermoplasty. So that's this treatment here. And what they do is they bring a catheter into the lung, and the tip here has electrical wires and you generate an RF field which heats up the tissue. And basically, with temperature, you're trying to damage that smooth muscle layer around the lumen. And the idea is if you damage it or you make it thinner, then the ability to contract becomes less. And so it will reduce the severity of an asthma attack for these patients. But when you and it turns out that you do this therapy for that is for severe asthma patients, and about half of them benefit from this therapy, and the other half don't. But it's unclear which patient benefits and which patient doesn't, and it's also very difficult to assess what the thickness of the muscle layer is before and after treatment. So what they do right now is they take a biopsy in this area. So they go in and they take a small biopsy at what you call the carina. That's where two airways split further. But it's also not known how representative that area is for the airway wall, where it's most important. So the idea is that you can use now OCT to go into that lumen and see if you can determine the thickness of the air of that airway smooth muscle. And so the concept is a catheter that rotates. So here you can see, it's a catheter with a diameter of 1.35 millimeters. It has an optical fiber. This is the optical fiber. This is a lens. It's a rod lens. We have an electrical motor at the tip. That is this one here. And we have a motor axle with a prism on top, and then the light is reflected outwards, and as a lighthouse, the beam rotates, and when we do a pullback of this catheter, we get a cross-sectional images and basically a whole volume. Okay, this is the actual catheter tip. What you can see here is this is the Grin lens. This here is the prism glued onto the motor axle, and this thing here is the motor. It's a hand-built motor in a machine shop. With here you see the electrical wires to put a current to create a magnetic field, and then the core of the motor is here, a magnet that is around an axle, and this thingy can then rotate at 50 to 200 rotations per second.
I have one question.
Yeah.
Maybe it's a bit of a stupid question, but I'm not sure if we do not want the muscles around the lumen to contract. Why do we have them?
Ah,
Like, what is their purpose normally?
good question. I don't know. Um, uh, it might be a function of your in and out breathing, right? Well, the main part is the muscles in your chest that make you expand your chest, and then you suck up the air, and then you can expand. If you it may be part of the process of getting rid of stuff that enters the lung. So if you cough, for instance, you try to get rid of stuff, but also it's mucus and other things. Um, but I'm just guessing.
Yeah, okay.
It's there and it's a problem, or in these patients. So, but what happened now? Oh, yeah, here. So here we look at this here is histology. So this is basically from an ex vivo lung. So it was for surgery or something, and they create histology of it. So this is this image, and then you can see the different layers in the lung. You can see the epithelial layer, the lamina propria, the basal membrane, and the airway smooth muscle. That is this here. This is the structure we're interested in, and so you can use a different stain in your histology, and then the airway smooth muscle really becomes apparent. So this is um, also histology, but this time it's colored with something that's called Desmin, and it colors all the muscle tissue brown. So here you can see now what the location is of that muscle around the lumen of the lung, and this is a fairly large airway. Now this is the standard structural OCT image that you are familiar with. When you look at the larger depth, you can see that the signal fades away due to the exponential decay of the signal. And so at the surface it's dark, that's a strong signal, and then at larger depth it becomes gray, or a lack of signal. And then what you see here is these structures. And we really were confused by these structures in the beginning. We had no idea what they were. But then we switched to the attenuation coefficient images. And now you can see sort of an outline of these thingies. They look like this. And when we go back to the histology, then we realize what it was. It's a fold of the tissue. Huh? So here you can see a fold in the tissue. So the lumen can be much larger, but when it collapses, you get a folding of the surface, and this is what we're looking at. So these folds here, this is another fold. That is what we're looking at here in these in the OCT images, and we think that with the attenuation coefficient, it's easier to identify. Okay, this is a movie. Can you see the movie? Yeah, it's on my screen. It's much better because it's like 20 frames per second, and for you, it's like, I don't know, much lower.
Yeah, about 2 to 3.
So the question is, how deep can this probe go into the lung? Actually, it can go all the way to the periphery. So you go in through the mouth, you go down, and then you take a left or a right branch, and then you can keep on going until you end up really somewhere very close to your chest wall here, or in the lower lobe, or you can go up and you get into upper lobe here. You can get very far, very far. Okay, so attenuation coefficient images. Let's look at another kind of structure. So here again, this is the standard H&E stain of the histology. And here again, we see the Desmin stain for muscle. And what we see here are cartilage rings. So this is cartilage. This is cartilage. Oh, it says collagen. I make a mistake. Nee, that must be cartilage. Now we can look at the OCT image. And what I did here is I switched from the standard to the attenuation coefficient image. So this is your standard image. This is an attenuation coefficient image. And what I would like you to see is this structure here. So this is the cartilage that you also see. Now I see it. Here it says cartilage. This is cartilage. This is cartilage, and the collagen is I think it's here. Anyway, you can see now this is if you look at my marking, this delineates the. Your screen doesn't update. That's unfortunate. Oh, now it does. So now you see the same outline that I drew in the attenuation coefficient. I show it in the standard image, standard OCT image, and I think that outline of the cartilage, it's much clearer in the attenuation coefficient versus the intensity image. So again, this is intensity, this is cartilage, and I can even try to remove my marking again so you can judge for yourself. Standard OCT image and the attenuation coefficient. Okay. So we can use now the polarization sensitivity to extract that airway smooth muscle layer. And this was work that was done by Adams. That's a group in Boston, Mass General Hospital, where they created their standard structural image. This is the segmentation of the airway smooth muscle based on polarization, and this is actually the histology of that same area. This is from an ex vivo pig bronchus. So now we want to go to the data of the in vivo asthma patients. So again, you're familiar now with the structure of these images: attenuation, birefringes, and optic axis uniformity. And this is now a complete pullback where we just show the optic axis uniformity. So we can see now, here we see this layer here, we see this layer here, layer here, and we can use that. Oh, it's rotating, it's beautiful. Does that come across, that this whole volume rotates?
Yeah, but a little slowly, of course.
Yeah, it's missing a lot of frames. But you can see now that over a length of 11 millimeters, we can determine areas of high fiber. It does not necessarily mean it's smooth muscle. It can also be collagen, but at least this highlights it. And the second image that I'm going to show you is where we try and come up with the orientation of that layer. So that is shown in these images. Here we try to determine what exactly is the orientation. And then we see, for instance, here, this blue orientation. This is what we think now the airway smooth muscle layer. And in the second one, we see it also, but here you can see it's more purple. So that's a problem of our measurements that we're fixing right now. We can measure an angle, but it has an offset, and we don't know what the offset is at the beginning. So a layer might look purple in one image and might look green in the other one, but the orientation of the fibers is the same, and they're all circular around the lumen. So actually we have more recent data where we were able to fix that, and then I need to. So I hope it's not okay. For me, it's a movie that runs at video rate. It's okay. This is a good example of here. You see that nice layer here, airway smooth muscle. Okay. So this is now the area smooth muscle as a percentage of the total area of our OCT cross section. And we do that in a patient in the exact same location before and after this treatment. So here, this is before treatment, and this one is after treatment. And what you can see is we can recognize the more or less the same structure in the sense of the thickness of that airway smooth muscle layer. It's a pattern that kind of reproduces, but only before it's much thicker than after. So this is our first measurement of the effect of this treatment in an 11 millimeter long section somewhere, I think right here, and then about 5 centimeters deep inside the lung. And then finally, we did this in 4 patients before and after. No, in 1 patient before and after, and in 3 other patients only after treatment. And we also measured collagen content by taking the biopsies. So that's the current standard method for the clinicians to determine the effect of the treatment. They just take small biopsies. So along the horizontal axis, you see the average percentage of airway smooth muscle determined from the biopsies, and we plotted that against our assessment of the airway smooth muscle by OCT. And you see we find a very nice linear relation between the two measurements, actually with a significance of 0.05, p-value of 0.05. So only 4 patients. It's not easy to do, but already significant. So we're extremely excited about these results. And that brings me basically to the end of my presentation. And this is Jouke Annema, a professor in the lung department. This is Peter Bonta. So these are the 2 clinicians. This is the PhD student, Annika. This is Margarita. She is my PhD student doing all these measurements, and this is me. Okay, with that, so that last part was kind of how can you use it and what's it good for? Because it's a complicated technology, but we think there is where it can be very useful. And that was my presentation. If you have questions.
Thank you for the lecture. It was really interesting.
All right. I think the 2 of you really pulled me through this 4 hours. It's so much better to have a face to look at when I'm talking and to get
Yeah, I can imagine.
Dankjewel, Joris.
Yeah, okay.
Thank you.
Bye.
Bye-bye.
Yeah, take care. Bye.